Hi, everyone, and good morning. After taking a break for a few weeks, we're back with today's Grand Rounds, and I'm really excited about this talk as we have Dr. Gregory Fonzo here from the University of Texas at Austin, who's going to be talking with us about his work on neuroimaging prediction of antidepressant treatment response. Dr. Fonzo graduated from the University of Georgia in 2005 with a dual degree in biology and psychology, and received his doctorate in clinical psychology in 2013 from the combined program San Diego State University and UCSD. He also completed two postdoctoral research fellowships where he was engaged in a T32 biobehavioral research fellowship, as well as a VA MyRec advanced fellowship in mental illness. He now runs his own lab in the Department of Psychiatry at UT Austin. And in the process, he's published several dozen papers in widely respected journals such as Nature, JAMA Psychiatry, and the American Journal of Psychiatry. Dr. Fonzo's work centers around mood, anxiety, and trauma-related disorders. And in particular, today, we're going to be reviewing some of his work about how we can best use neuroimaging to not only understand the mechanisms at play for our patients, but also to improve our clinical care. This is in large part why I find Dr. Fonzo's work so fascinating. It takes the science of discovering underlying mechanisms and translates it into practical, real-world situations that we all encounter when working with our patients every day. I personally have greatly benefited from reading Dr. Fonzo's research, and I'm really excited that he's here today to talk with us, and I'm hopeful that everyone will be able to gain some helpful insights from today's talk. If you have any questions for Dr. Fonzo, don't forget that you can use the Q&A button to submit it at any time. After the talk, a survey should open that you can complete to claim CME credit, and if the survey doesn't appear, you should get an email with the link to the survey tomorrow around noon. And now I will turn it over to Dr. Fonzo.
Excellent. Thank you, Jonathan, for that great introduction. So yeah, I'm excited to be here today to talk with you and present on neuroimaging prediction of antidepressant treatment response. And oops, here we go. Just really quick, the learning objectives for today's talk are going to be for you to recognize the heterogeneity in major depressive disorder and the effect that this has on antidepressant responses. Secondly, identify neurobiological characteristics that predict antidepressant response in depression. And identify key strategies and considerations for the development of predictive biomarkers. And I thought I would begin this talk by talking a little bit about a true story of a clinical illustration of kind of the need for this type of work and technology that first got me thinking about this. So I was a graduate student at UC San Diego, working as a practicum student at UCSD Outpatient Psychiatric Services, and began to see a patient named Christina, which we'll call her in this context. And she was a 23-year-old female, first-generation Asian American, with a history of severe major depression, social anxiety, and borderline personality traits. Now, Christina was seeing me under the guise of therapy for academic problems, but in actuality, she was really severely depressed. And that was certainly, I think, one of the primary issues, but also contributing to her academic difficulties. And the reason she had this guise of seeing me for academic problems was related to a stigma that she felt around being depressed. And in her family and in her culture, it was sort of viewed as a form of weakness or something that was generally looked down upon. And so that was one of the reasons for that academic problem presenting issue. Now, she had tried many different antidepressants in the past. I couldn't even remember all of them, but none of them had really worked for her, unfortunately. So we tried a course of psychotherapy, primarily cognitive behavioral therapy, teaching her how to reappraise negative thoughts and emotions, some behavioral activation, and then building in some DBT skills as well, such as mindfulness and emotion regulation to help her deal with some of her distressing emotions. But a big part of what came up in that was the discussion around the stigma that she felt for having depression and other and anxiety and other forms of psychopathology. And I was able to use a lot of psychoeducation in regards to some of the neurobiological bases of depression and some of the biological and hereditary predictors of depression to help her to alleviate some of that stigma and to help her realize that in actuality it was not a form of weakness on her part. And in that process, it got me thinking a lot about the neurobiology of depression and whether it might be possible to begin to use measurements of this neurobiology to predict whether somebody might be a responder to medication or, for example, something like CBT, which would have certainly benefited Christina in this context in terms of eliminating a lot of the needless medication trials that she had to go through. And this is a key element of personalized psychiatry, which is basically tailoring the treatment to the individual and trying to make that as most effective and efficient as possible. So a little bit about depression. We all know it's a major public health problem, but just to illustrate this is a table from a publication from 2011 showing the different prevalence rates of depression across various highly developed countries. And you can see here that the lifetime prevalence of depression on average is about 15%, and about 20% in the US. So over the course of a person's lifetime, about 1 in 5 will experience major depression, which is quite a high number. Additionally, the 12-month prevalence rate any given year ranges about 6% on average, but anywhere from about 5 to 9% across these different countries. So this is definitely a major public health problem that many of us are likely to either experience or witness in our lifetime. And the one thing about depression is that it is very heterogeneous. It comes in many different forms and flavors. For example, the diagnostic criteria we have nowadays, you need 5 of 9 symptoms to reach an MDD, major depressive disorder diagnosis. And that alone gives us 227 different combinations of symptoms that an individual could have in order to meet that diagnostic criteria. Now, if we include the symptoms that include a sort of this bipolar element of either increased or decreased sleep, appetite, or movement, that gives us 1,497 different combinations. So as you can see, stating that somebody has depression is a little bit like stating somebody has a car. You know, it doesn't tell us much about the type of car, how it drives, and the various characteristics. And yet those individuals typically all get lumped together in one category. And this doesn't even include the various subtypes and descriptors of depression that we also have in our diagnostic system. Now, part of the some of the driver, at least of the heterogeneity in depression, undoubtedly relates to genetics. And we know that there are many different genes and genetic characteristics that predict independently a risk for major depression. So publications have found 87 different risk variants with 269 individual genes, for example, that make one more susceptible to developing depression. And these genes influence a wide range of mechanisms, including things like calcium signaling, dopamine signaling, and glutamate signaling in the brain. So it's quite feasible there are many different forms of depression that underlie or underlaid by these different genetic factors. In addition to that, we know that the brain is also heavily shaped by early life experiences, and things such as child abuse, child maltreatment, and early life adversity also undoubtedly play a role in predisposing individuals to this disorder. Now, I've talked about depression, but I haven't even mentioned the comorbidity. And what we do know is that depression is highly comorbid with anxiety disorders, PTSD, obsessive compulsive disorder, and ADHD. Those are some of the most frequent. And that we know altogether about 72% of people with major depression will also present with some form of comorbidity. So altogether, depression, it's a widely heterogeneous category. And this heterogeneity also contributes to heterogeneity in responses to antidepressants. So on average, the meta-analyses tell us that the effect size for an antidepressant versus placebo is about 0.3 on average, which is fairly small. And for example, in STAR-D, which was a big study looking at sequencing of depression treatments, only about 30% of individuals remitted on the first treatment try, and that was with a common selective serotonin reuptake inhibitor, citalopram. Another 30% of individuals in that study did not remit after 4 consecutive treatment courses. So that tells us something about the way in which there's a great deal of heterogeneity in response to how somebody might benefit from a particular antidepressant, or several in this case. And this has even led some to argue that antidepressants don't work at all, and they're simply a powerful placebo. I would take a different view, a more balanced one, I guess, in that I would say that despite these kind of sobering statistics, that some individuals will definitely benefit from an antidepressant. But it's simply a matter of identifying who that individual is and funneling them to the right treatment. Now, several studies have looked at predictors of antidepressant response, particularly clinical and demographic. And so, for example, we know that anxious depression is a poor predictor of antidepressant treatment response. Various demographic factors also contribute, like gender, ethnicity, socioeconomic status, and education. And then a depression severity level at baseline can also be a significant moderator of antidepressant response. However, alone, each of these accounts for only about 5 to 10% of the variance in outcome. So they're relatively small or weak predictors of antidepressant response. They're also sparsely replicated in the literature, so we don't have a lot of evidence that these things will hold up across multiple samples. And these clinical and demographic factors also give us a little information on mechanisms of depression that might underlie some of the symptoms, as well as potential modification targets that we might be able to change in order to make somebody more likely to respond to an antidepressant. So this really motivates the perspective that if we can assay something that is closer, ostensibly to the mechanism of depression, which we believe is a brain disorder, then we might be able to more effectively predict how people will respond to antidepressant treatment. So for example, using non-invasive tools like MRI or electrophysiology, we might be able to develop predictive biomarkers that we can then apply to our heterogeneous population and then hopefully identify who's most likely to respond to a given treatment, such as an antidepressant or cognitive behavioral therapy. Now, let's talk a little bit about the functional neuroanatomy of MDD, just to give you a brief overview of the literature, which is quite broad and varied, but this will help you contextualize some of the later topics that we discuss. So depression frequently has implicated the amygdala as being an important part of the abnormalities in the brain relating to the symptoms. We know the amygdala is heavily involved in fear and threat processing, as well as reward processing. So it seems to serve sort of a bivalent salience detection effect. Likewise, the insular cortex has also been heavily implicated in depression. And the insula is heavily involved in a construct known as interoception, or sensing the overall physiological condition of the body at a given moment in time. Now, these body state representations are successively re-represented in the insula over time. And as such, they play an important role in things such as emotion, but also cognitive processing and cognition, as well as sensory and pain processing. The hippocampus in the medial temporal lobe is also heavily implicated in depression, and we know that this is a brain region that's very important for memory processing as well as learning. Now, in the prefrontal cortex, many of us have probably heard of the subgenual cingulate, and this is a portion of the brain that is heavily important in the visceral or autonomic aspects of emotion, and one that has been repeatedly implicated in a part of the pathophysiology of major depression. Further anterior in the brain, we have the ventral or pregenual cingulate. And this is an area of the brain that is involved in decision-making, reward processing, and also the implicit regulation of emotion, which is a topic we'll discuss a little bit later on as well. Now, more superior in the brain, we have the dorsal cingulate, which is heavily involved in conflict and cognitive processing, as well as emotional reactivity. And this is another area that is frequently implicated in depression. And then finally, subcortically, again, we have the ventral striatum and the nucleus accumbens. And this is a portion of the brain that is critically involved in reward and pleasure processing, as well as motivation. And so these are all areas that you will see presented in some of the subsequent studies that we'll review. Now, on the lateral side of the brain, to continue with this, we also have the dorsolateral prefrontal cortex, which is also heavily implicated in depression. And this is an area of the brain that's involved in a lot of higher order cognitive processes in humans, things such as executive function, as well as cognitive reappraisal and deliberate regulation of emotional state. And then more superior, we have the more inferior, we have the orbitofrontal cortex, which is a portion of the brain that's involved in reward processing and representing affective value, so the value of a stimulus in terms of an emotional response. It's also heavily involved in pleasure processing and frustrative non-reward, or irritability might be another word for that. So now that we've reviewed some of the functional neuroanatomy, let's turn to looking at some of the brain function abnormalities that have been observed in major depression. And much like the clinical manifestations, the brain abnormalities that we have seen in depression are also heterogeneous. For example, some meta-analyses have failed to converge on any consistent abnormalities across the disorder. However, others have isolated consistent effects, and a lot of this probably depends on the literature that was surveyed as well as the methodology utilized. But I'll just briefly review some of these abnormalities and break them up by what we'll call cross-domain or sort of general abnormalities observed across many different psychological processes, those involved in negative emotion, those involved in positive emotion and reward, those involved in cognitive processing, and then resting state connectivity, which is a very a widely utilized metric of brain network function. So first, let's begin with the cross-domain abnormalities, or those that we have seen across many different psychological processes. And so a recent meta-analysis in 2020 observed that the hippocampus on the left side, the right amygdala and putamen, and the subgenual cingulate all displayed increased activation in major depression, as well as diminished gray matter volumes, which is a measure of structural brain integrity. Now, for negative emotion, we've kind of see in major depression this increased activation of this triad, we might call it, of the amygdala, insula, and dorsal cingulate. And so you can see here a 2012 meta-analysis identified increased activation in major depression for all three regions, amygdala, insula, and dorsal cingulate. And then a later meta-analysis in 2013 also convergently observed abnormalities in the dorsal cingulate and in the amygdala in major depression, particularly increased activation there relative to healthy controls. Now, on the other side, we tend to see decreased activation during negative emotion processing in the dorsolateral prefrontal cortex. And so the same 2012 meta-analysis depicts it here in blue. And same thing is seen in the 2013 meta-analysis, where we can see decreased activation in the dorsolateral prefrontal cortex. Now, turning to positive emotion or reward processing, it's another heavily studied area in depression. What we tend to see across studies is decreased reactivity of the ventral striatum. You can see here a 2019 meta-analysis observed decreased activity in this ventral portion of the striatum during reward processing in depression. And same thing was observed in a 2013 meta-analysis in a slightly more posterior area in the ventral striatum. Now, on the other hand, we also tend to see increased activation in depression in the orbitofrontal cortex during reward processing. You can see that depicted here in yellow in this 2019 meta-analysis, and here in red in the 2013 meta-analysis. So this seems to be a pretty consistent abnormality in the reward circuit related to depression. And then when we looked at cognitive processing, what we see is, in general, we see decreased activity of the insula cortex, here shown in a 2012 meta-analysis in depression, and then here also shown in a 2017 very large meta-analysis across all of psychopathology, displaying decreased insula activity during cognitive processing. Likewise, we also tend to see decreased activity in the dorsal and pregenual cingulate, here depicted in a 2012 depression-specific meta-analysis, as well as, again, in this 2017 general psychopathology meta-analysis of cognitive abnormalities. Now, resting state has also been heavily examined in depression, and a paper from 2015 put forth a network model that kind of related a lot of these findings. And there's a few features I'd like to point out here that I think will be helpful. One is that depression tends to be characterized by decreased connectivity in this frontoparietal control network, which is here depicted in green. And this is a network that's heavily involved in top-down control, executive processes, and a lot of sort of cognitive processes. And so that's kind of consistent with some of the cognitive difficulties we might observe in depression. Likewise, we tend to see increased connectivity between this frontoparietal network and this area here, or this network here called the default mode network, here depicted in red. And then the default mode network is heavily implicated in self-focused processes, and it's thought to underlie some of the ruminative manifestations of depression. And consistent with that, we also observe in depression increased connectivity within this default mode network. Now, before we begin to talk about predictors of antidepressant response, neurobiological predictors, let's discuss a little bit about some of the changes that have been observed in the brain related to antidepressants, which I think will help to contextualize some of the predictive findings. And we have seen that antidepressants do tend to decrease amygdala and insula brain responses, particularly to emotional stimuli. And we see that both in depressed populations as well as healthy participants. And interestingly, this is an effect that is observed, at least in healthy folks, even with just a single dose of an antidepressant. So that's quite an interesting finding that suggests that this brain signature of antidepressant may or may not be part of the therapeutic effect, given that it happens so quickly, and given that antidepressants typically take several weeks before we see an effect. But this is begin to or before actually I discuss the next part, an additional aspect to this too is that antidepressants tend to increase brain responses to positive stimuli. So we see a general decrease to negative emotional stimuli and a general increase in brain response to positive stimuli. And together, this has kind of led to this model that has been put forth by Catherine Harmer and others, which basically relates the delay in antidepressant effects to a specific mechanistic process, whereby the serotonin increase that is affected by the antidepressant is thought to create some downstream neuroadaptive effects in the brain, which we might be seeing here, that leads to a change in emotional bias, whereby individuals become less emotionally biased to negative stimuli and begin to pay more attention to positive emotional stimuli as well. And the idea is that over time and with repeated experiences of that, individuals begin to learn, a learning process occurs, and that leads to an improvement in mood and an alleviation of some of the depressive symptoms. So if we look at brain prediction of antidepressant response, there's been several studies that have been published on this point. And first off, what we have seen from this study known as iSpot-D, which stands for the International Study for the Prediction of Optimized Treatments in Depression, is that individuals that show less amygdala activation to an emotional stimulus prior to undergoing treatment tend to do better with antidepressants. You can see here, the R stands for responders to antidepressants, non-responders is NR, and then here are healthy controls. And one pattern that I'd like to point out here too is that healthy controls seem to fall in between responders and non-responders in terms of the level of brain response in these areas. And that's something that I think you'll see play out again and again in these findings that will be important to note. Now, in terms of another predictor that's been observed is increased activity in the dorsolateral prefrontal cortex, specifically to a cognitive process or a cognitive task that provokes inhibition. And we can see here that remitters show greater activation relative to non-remitters from iSpot-D. And then this has also been investigated in the context of resting state connectivity. And what we can see here is it's a complex pattern of effects, but one thing to point out here is that a greater connectivity within the default mode network and greater connectivity between the default mode and the frontal parietal network tended to predict a more favorable response to antidepressants. And here is sort of the average across all of these network connections. Again, with healthy controls being somewhere between remitters and non-remitters in major depression. And 2018 paper also observed the same effect, specifically within the default mode network. So greater connectivity at baseline predicted more favorable response to an antidepressant. And so this begins to paint the picture of that the brain phenotype that might be best suited to responding to an antidepressant is able to regulate emotional responses, as we can see from this study from Lee Williams, be able to deploy prefrontal resources when necessary in terms of a cognitive processing paradigm, and also be able to maintain a network connectivity between various networks at rest. Now, there are certain key limitations to the literature I just reviewed, and one of the main ones being that all those effects were simply predictive, meaning that they predicted a response just within a single group. And ideally, what we want to do is have a moderation effect where we look for a differential prediction between two or more groups. And that allows us to begin to control for some of the other non-specific effects of an antidepressant, like the placebo response, for example. And that gets to the second point in terms of a lack of placebo control. Thirdly, there were really no biological probes in those studies of certain processes that we know are particularly important in major depression. One of those would be emotion regulation. And we know that is an area that is heavily studied in depression and is known to be abnormal. And the other would be reward processing in terms of how people respond to and learn from positive stimuli. And we know that both are abnormal in major depressive disorder, and both have been found to change with antidepressant treatment. So this suggests that these two constructs in particular may be fruitful for yielding a antidepressant predictive effect. And so the study that most recently came about to address some of these limitations was known as EMBARC, which stands for Establishing Moderators and Biosignatures of Antidepressant Responses for Clinical Care, kind of a long name. But it was quite a large study. 309 individuals were randomized to sertraline versus placebo. They had neuroimaging at baseline prior to undergoing treatment, and then 1 week afterwards. And in terms of the imaging tasks that were utilized, one was an emotional conflict task, which is the one I worked on, which I'll present in a little bit, some of the results from that. We also looked at resting state connectivity in these individuals, and then they also had a reward processing paradigm in order to get at some of those constructs we discussed earlier. And the goal of the study really was to establish moderators and mediators of antidepressant response. So that is, what are baseline neurobiological characteristics that differentially predict a positive response for an antidepressant versus a placebo? And then what, if any, are changes from baseline to 1 week that might underlie some of the, or relate to the subsequent therapeutic effect of antidepressant versus placebo, which would be the mediator. And so we talked about emotion regulation as being an important construct, and I'd like to take the moment to just discuss a little bit about emotion regulation and sort of the idea of it being a dual process, which will help to understand some of the EMBARC work. So my old mentor, Mead Etkin, had proposed that emotion regulation really exists at least in 2 forms. One would be an explicit regulation process, and this is the type that most of us are familiar with, and the type we might teach people in therapy, for example, in terms of how to cognitively reappraise an emotion or a situation. And this tends to recruit areas of the brain, such as the dorsolateral prefrontal cortex, parietal cortex, and then areas of the dorsomedial prefrontal cortex as well. And so explicit regulation is something that's deliberate and effortful. It's something that the person is aware that they're doing. However, on the other hand, it's been proposed that there is also an implicit regulatory process in the brain that's responsible for regulating emotions. And that area, or the part of the brain that's specifically implicated in that, is the ventral cingulate and the ventromedial prefrontal cortex. And this is akin to a process that kind of occurs in the background, is automatic and non-conscious and non-deliberate. So it's something that is sort of going on at much of the time. And the way I kind of think about it is sort of like the shock absorbers on a car. They are there to simply kind of smooth out the ride. And we know from various studies that Dr. Etkin has done that the ventral cingulate in particular is abnormal in depression. And this is particularly relevant for predicting antidepressant response, because we know that the ventral cingulate and amygdala pathway, for example, is heavily modulated by serotonin. And so we might expect that a serotonergic antidepressant would yield, or that the function of the circuit at baseline might thereby predict responses to that treatment. And so in EMBARC, we had a probe of this implicit regulatory process that is called emotional conflict regulation, which occurs in the context of this emotional conflict task. And the way this test works is individuals are presented with successive trials of emotional faces, either fearful or happy. And then there's an emotion word written across the face, which is either congruent or incongruent with the emotional expression. And they're instructed to simply identify the emotion of the face and ignore the overlaid emotional word. Now, when the word is incongruent with the facial expression, this causes a conflict effect whereby individuals slow down in their reaction times, and they have more difficulty identifying the facial affect and ignoring the emotion word. However, when that incongruent trial follows another incongruent trial, there's a conflict regulation effect that occurs, whereby on the second trial, individuals actually mitigate their reaction time slowdown, the conflict effect is decreased, and they are better able to identify the emotional expression. So there's a sort of an adaptive effect that occurs from trial to trial in this paradigm, and which has been kind of well characterized. And we know from prior work that this conflict regulation effect heavily recruits the ventral cingulate and the ventromedial prefrontal cortex. So we tend to see increased activation there on the second conflict trial that is follows the first one. And we also tend to see decreased activity or dampening of activation in the dorsolateral and ventrolateral prefrontal cortex, as well as the insula, which are conflict responsive areas that are typically activated during the conflict processing of this task. And what we know from prior work is that this ventromedial or this ventral cingulate response to conflict regulation is typically abnormal across not only depression, but also generalized anxiety disorder and their comorbidity. So it seems to be indicative of a common circuit disruption across these mood and anxiety conditions. And likewise, in conjunction with that deficient ventromedial prefrontal response, we also see a lack of an inhibition of the amygdala when individuals are adapting or regulating emotional conflict. And that's also similarly seen across generalized anxiety, depression, and their comorbidity. So this was our probe of emotional conflict, or this implicit regulation process in EMBARC. And now, before we turn to the predictive effects, just to briefly present the clinical findings, what we see, as expected, is sort of just a very modest benefit of sertraline over placebo. So this is time in weeks, and then HAM-D17 depression severity rating on the y-axis. And we can see that those with sertraline on average across all individuals tended to do slightly better relative to placebo, which is consistent with the literature. Now, when we looked at the baseline brain activity and tried to use that as a moderator of antidepressant response, what we found is that there was a very widespread moderation effect across the brain. You can see here on the cortical surface, the yellow indicates voxels that were significant moderators of the antidepressant versus placebo effect. And then you can see here as well, we detected some areas in subcortical circuitry. And when we looked to break this down a bit to understand what this moderation effect was showing us, what we saw is that in areas that are typically conflict responsive, so we might call these the conflict response network, areas such as the dorsal cingulate, the dorsolateral or frontopolar cortex, and the anterior insula, that individuals here below the median level of activation tended to do better with antidepressants. So the way to read this is that this is a median split of the activity in this particular area. And individuals here are those with depression that are below the median response and above the median response. And then healthy controls are in the leftmost quadrant. And what we can see is that, like I showed before, that healthy individuals tend to fall in between those that did better and those that did worse relative with in major depression with an antidepressant. And so we can see is that those below the median level of activation, i.e. those that better dampened activity in these regions on that second conflict trial, tended to show a much more favorable response to an antidepressant, here in blue, sertraline, versus the placebo in red. And you can see those that did not effectively dampen this conflict responsive network showed really no benefit of the sertraline versus placebo effect. So let's turn now to looking at the reward circuit and what we found in EMBARC in relation to predictive effects. And so in EMBARC, there was a reward paradigm utilized, which is illustrated here, that I'll briefly explain. And basically, the individuals on every trial had to guess at the beginning whether a subsequent number was going to be higher or lower than the number 5. And so they had a 50-50 chance. It was 1 to 4, and then 6 to 9 on the other end. And so first they made a guess as to whether it'd be higher or lower. Then they saw this expectancy queue in which they were told they could either win money on that trial if they got it right, or if they didn't get it right, just not win any money, so no loss. Or on the other hand, if they saw a down arrow, they could either lose money on that trial if they did not get it right, or not lose any money if they did get it right. So it had sort of this both win and loss component built in. They then saw the number presented and then were presented with feedback about whether they won, lost, or did not change in their winnings. And so if they won $1, and if they lost 50 cents. And so what the authors did with this paradigm is they looked at a form of reinforcement learning that is exemplified by what's called a temporal difference model. And the idea here is that the ventral striatum in particular is thought to track a reward expectancy signal that adheres to this temporal difference model. And the way this works is across time, individuals see the reward expectancy cue, and then they learn over time that that cue is associated with the later reward response. Now, what happens is with successive learning opportunities that this learning that occurs in the outcome phase begins to feed back to the expectancy phase, and individuals begin to associate the expectancy cue with a reward, and they show less ventral striatal response to the actual rewarding outcome itself. So that is what is predicted by this temporal difference model of reward learning. And so the authors attempted to look at the degree to which the ventral striatum adhered to this temporal difference learning model and how that might predict antidepressant response. And so they incorporated a reward expectancy cue or regressor at this point in the task. And it was modeled as plus 0.5 if they saw the up arrow, which means simply the 50% chance of winning $1. So just the product of the probability and the reward magnitude. And then they modeled it as negative 0.25 if they saw a down arrow, which means 50% chance of losing 50 cents. They also incorporated a prediction error regressor here at the time of outcome, which if they saw an up arrow presented and they received money, they modeled that as 0.5. So the idea is they expected 50 cents from here, but they got $1. And so that's simply the difference between the two. Now, if they saw the up arrow, but did not win money on that trial, then they modeled that as negative 0.5, meaning they expected 50 cents, but got $0. Similarly, if they saw the down arrow first, meaning a potential loss, but they avoided the loss, then they received, or they modeled this as 0.25, where they expected a loss of 25 cents, but got 0. And then similarly, if they had a potential loss and then they did receive the loss, they modeled that as negative 0.25. Okay, so what the authors did then is they constructed this reward index of the ventral striatal response to get at this temporal difference learning model. And so what they did is they looked at the reward expectancy ventral striatal response during the second half of the task minus the first half. And the idea here is that the response, if it adheres to this model, should be larger in the second half of the task and smaller in the first half. And similarly, they added that to the difference between the prediction error response during the first half of the task versus the prediction error response in the second half of the task, which should ideally be the opposite of this one, larger in the beginning, and then it gradually gets smaller as the task goes on. And so the idea here is that individuals that show more positive reward index values adhere more to this temporal difference learning model, whereas those who show more negative values are the most divergent from the learning model. And what they found is when they use this learning model to try and moderate treatment response to antidepressants, is they found that individuals that showed a more negative reward index tended to do better, meaning that individuals that had more of a divergent ventral striatal response that did not adhere to this temporal difference learning model, those people tended to do the best with an antidepressant. Now, when they split this up and they looked at the different contributions of reward expectancy and prediction error, what they found is that it was really the reward expectancy signal that was providing the predictive effect. And you can see that here on the bottom row. This is the reward expectancy subindex showing a moderation effect. Here is the individuals 1 standard deviation below the mean, at the mean, and then those above the mean. And you can see that the difference in responses is greatest for those who were 1 standard deviation below the mean. So this gives us some indication about the degree to which the ventral striatum might be predictive of antidepressant response. And it still remains, there's still some unanswered questions with this study in terms of whether it was the second half reward expectancy response or the first half that was really contributing, or some combination of the two. But it does give us a starting point to begin to investigate these questions in future studies. Now, finally, we'll talk a little bit about the resting state connectivity predictors or moderators that we observed in EMBARC. And so this was a paper published just recently in 2020 by Shirish Chinfat. And what you can see is that this predicts the different networks of the brain, the resting state networks, and the thickness of the line indicates the amount of connections between those networks that were moderators of antidepressant response versus placebo. And you can see that there's quite a bit here. So it's kind of a lot to digest. So what they did is they sort of noted these down excuse me. What you can see here is that they broke this up into those that predicted sertraline specifically and those that predicted placebo. And one thing to note here is that this is the sertraline predictive effect. Top row is areas that had greater connectivity, predicting a better response to sertraline versus placebo. And then those on the bottom are those that showed less connectivity, that showed a better response. And one thing to note here is that you can see that connectivity within the default mode network in particular was a robust moderator of antidepressant response, such that those who had a greater level of connectivity at baseline tended to do better with antidepressant sertraline versus placebo. Okay, so just to give a brief interim summary, we've seen that better antidepressant response has been moderated by, or in this case, better dampening of conflict responsive regions during implicit regulation of emotion, ventral striatal activity that is less adherent or more divergent from this temporal difference learning theory model, and in particular, greater connectivity within the default mode network and greater connectivity between the default mode and the executive control network, here depicted, also predicted a more favorable response to antidepressants. And so this sort of set of findings got me thinking about how do we conceptualize, the dimensions along which we might be able to predict antidepressant response. And so a priori, I was sort of thinking about it in a kind of unidimensional way, where if we think about, this is a healthy brain and this is a depressed brain, that maybe individuals that are depressed that fall either closer to the healthy brain or closer to the canonical depressed brain might show a more robust predictive effect to antidepressants. But in looking at some of these findings, I think we have to sort of question that idea. So what we can see, for example, in a lot of these is that the controls typically tend to fall somewhere in between the spectrum of depressive brain activity measurements. And so, we can see it here and here as well, and then also in this study from iSpot-D. And so this begins to set up, I think, a different model for how we might think about this. And I think the way I'm thinking about it now is that we might think about healthy folks as occupying perhaps a certain part of a center part of the spectrum, and that we possibly have different dimensions along which antidepressant response might be predictive. And these dimensions could represent many different things. It could be various types of symptomatology or manifestations of depression. It could be a specific circuitry characteristics that are abnormal in depression. But I think this way of thinking about it as a multidimensional space is probably going to be the most effective way to try and conceptualize how we might predict antidepressant response. And particularly the idea that individuals, the predictors for these different dimensions could be different, or they could be similar. So I think it kind of sets up a different way to sort of approach this problem. And then related to this, I wanted to talk a little bit about a group versus individual level prediction, which I think is an important thing to discuss here. And so I think pretty much everything I presented up until this point has looked at predicting a group level effect, meaning that we have this group of heterogeneous individuals, we look at some factor, and we see the degree to which that factor might differentially predict to a response to an antidepressant versus, say, a placebo. And though we call this a sort of moderation approach. And what we get is some difference in symptom change trajectories as a function of different levels of this factor. Now, this does have certain pros, including the fact that it's typically easily interpretable, and we can, kind of isolate this factor and understand what it means. It's also computationally tractable, and it does allow for differential prediction, meaning that we can predict for one versus another, which allows us to control for some aspects of non-therapeutic or non-specific effects like placebo. On the other hand, there are certain drawbacks, which are that these effects are typically weakly predictive, only accounting for about 5 to 10% of the variance. It doesn't inform on individual treatment that well. So any one individual that may fall somewhere on the spectrum, it may not be a very useful characteristic for predicting that particular individual's response to an antidepressant. And then lastly, there's typically no assessment of the generalizability of these models, in that they're usually run on whole groups, and that there's typically very rarely a replication of the effect. So on the other hand, there's a complementary approach that is now being widely utilized in medicine, which is this individual level prediction, in which we take multiple factors now across an individual. And what we do is we try to come up with some complex weighting of these factors that allows us to predict the response to, say, an antidepressant, anywhere along the spectrum from good to, maybe not so good, or some binary split between these two. And this approach does have several significant pros, including the fact that it can inform on the individual level. So we can design a model that we might then be able to apply to an individual in the clinic, which will give us some indication or some measurement of the degree to which they're likely to respond to an antidepressant. It can also account for much more variance in the clinical outcome. And it also typically has a built-in assessment of generalizability, and that's typically some form of cross-validation. Now, on the other side, there are some cons to this approach, which is that it's typically very difficult to interpret what this complex weighting of factors actually means. And so that does make it more opaque to kind of understanding mechanisms and potential sources of modification. It's also computationally intensive and harder to control for non-specific effects. For example, for only predicting outcome to one drug, we don't know if the model itself is predicting a placebo response or simply something that's specific to the chemical itself. And then finally, these approaches are also prone to overfitting since we're combining across so many different factors. So some of these approaches have been applied recently, and I'll just highlight some of them here in EMBARC. And so this was the work of my colleague Yu Zhang. We sort of teamed up on this for the emotional conflict task data. And what he did is we took the brain images for each individual, looking at baseline emotion conflict regulation activation. And then we applied this parcellation across the whole brain, where we took average activation in each parcel across many different areas of the brain. He then utilized a technique called sparse Bayesian learning to predict response to sertraline. And here you can see the sparse weights are displayed on this brain surface here. And again, the full model is a complex weighting of all these different brain areas weighted by the various sparse weights. We then did a 10-fold cross-validation to assess the generalizability of the prediction. And what we found is that we were able to predict about 25% of the variance in outcome in EMBARC with utilizing emotional conflict regulation activation in this machine learning framework. Now, likewise, to assess whether it was specific to sertraline, what we did is we took the sertraline model and we applied it to data from placebo. And we found that we were really grossly unable to predict placebo response with this model, which suggests that the model may in fact be predicting something specific to the sertraline itself and not something that's a general characteristic of taking a medication. One issue with this, however, is that it's at this point quite unlikely that fMRI would be utilized on the individual level. So for one, we know that it's quite expensive. It costs about $600 per hour. It's not appropriate for everybody. So if you have metal in your head, we would not want to put you inside the MR scanner. And it's also typically not readily available in clinical settings. However, what we ideally want to do is find a measure of the brain that is convergent with MRI, but that is also more easily acquirable in these clinical settings. And so a good contender for this is electroencephalogram, or EEG, which we'll call for short, which is much, much cheaper to acquire, can be utilized on a broader segment of the population, and is typically much more easily implementable in clinical settings. And so we looked at EEG data in EMBARC, which was also collected, and this is the work of my colleague Wei Wu. And what he did is he took each individual's EEG at baseline, which is simply a measure of their brain at rest. And then he designed this really neat algorithm for basically designing a subject-specific spatial filtering approach, which basically allowed him to extract latent features from each individual's EEG data and then use those features in order to come up with a complex weighting to predict treatment outcome. And this paper is in Nature Biotechnology for any that are interested. And so I'm probably not doing it justice with this explanation, but here's an example of the latent signals that were most positive weights and most negative weights, and then also predicted here on the cortical surface to give you some idea of the contributors in the brain to this. And what he found is using this approach, this latent or sparse latent approach, is that he was able to predict about 36% of the variance in treatment outcome for sertraline data tested on sertraline. And then similarly, when he took the sertraline trained model and applied it to placebo, he was similarly unable to predict placebo response, which is also potentially indicative of a sertraline specific effect. And then finally, which I think is quite fascinating, is that we took the fMRI prediction model that I just presented and combined it with this EEG prediction model. And we looked at the degree to which the EEG prediction and the fMRI prediction actually related to one another. And what we found is that we did see a significant correlation between them, meaning that individuals that were more likely to be predicted to respond by EEG also tended to be more likely to respond as predicted by fMRI, which is a nice cross-modality convergence that begins to give us some indication that what we're measuring here is actually a coherent phenomena that's present in a particular individual. So briefly, just want to go over the take-home messages. MDD, or major depression, is a heterogeneous disorder, both in neurobiology and in clinical manifestations. Antidepressants are effective for at least a subgroup of individuals with major depression. Neuroimaging provides a neurobiological readout with some predictive validity for predicting antidepressant treatment response. And the informative features, I think, are varied and are likely to exist along several dimensions, which remain to be explored in future studies. And along those lines, I think the future research imperatives to point out here would be that we need to demonstrate the replicability and generalizability of some of these findings in order to really validate them, and then also demonstrate the incremental clinical utility, which would ideally be that, we could show that, for example, an EEG informed measure of antidepressant response might be useful in a clinical setting. I think another challenge here is we need to balance the transparency of the group level approach, which gives us very interpretable data, versus the approaches that are very complex, which are typically the individual level predictions. So there's an inherent trade-off there in terms of the transparency and complexity, but I think both are important to developing biomarkers in this field. And then third, we need to begin to meaningfully parse the heterogeneity in depression and use that parsing to be able to refine antidepressant predictive models. One way to do this is once we identify potential important features that might characterize the heterogeneities, that we can begin to build in model priors in a sort of Bayesian framework that begins to account for some of this heterogeneity, and then therefore will provide more robust predictions for treatment response. So I just wanted to briefly thank my collaborators and mentors. So particularly Meet Etkin, my postdoctoral mentor at Stanford, Yu Zhang and Wei Wu, who are colleagues that I worked with over there on my postdoc, and then Madhukar Trivedi for his facilitation of access to all of the EMBARC data. And then lastly, I want to thank the various sources of funding agencies that I'm currently working with. So thank you so much, and I'm happy to answer any questions or hear comments.
Yeah, thank you so much for going over that with us. We have a few questions that have come in. Actually, one question I had on my end is, having done the work that in this area, is there anything that's changed in terms of your clinical practice? Like, are there specific questions that you ask patients that, even though you're not putting them in an MRI machine like that, kind of get you towards thinking about how you might integrate this into your practice?
Yeah, it's a great question. I would say that no is probably the short answer, unfortunately. Yeah, I think at this point, it's still we're still too early in the translation of these into clinically useful metrics. But I think one way to do that potentially would be to utilize some of these machine learning approaches to try and relate, for example, a predictive brain effect to something that could be measured more easily in the clinic, like maybe a battery of self-report measures or behavioral measures.
Okay. And we had another question that came in. I think this was actually before you talked specifically about some of about EEG, but they were asking to compare the effects of what you found to other types of studies that you could do on this, like, for example, quantitative EEG or the dexamethasone's depression test, and how your findings compare to those.
Yeah, excellent question. Sorry, I don't know if I have an echo there. Yeah, those are, ideally, I think that's what we need, is to be able to relate across these different levels of analysis. And I think we kind of started to do that a little bit in EMBARC with relating the EEG and the fMRI. But ideally, we'd be able to get to something even more potentially quick to administer and cheap to administer, like something like a dexamethasone suppression test, which I unfortunately can't speak to in terms of how that might actually relate to some of these things.
Got it. Okay. Another question, I think kind of a clarifying question. Somebody had asked, the people who did better on antidepressants were less adaptive in their reward expectancy. So does this suggest that they were more optimistic?
Yeah, it's a good question. In that study, they didn't relate that reward index to any sort of measure of, for example, optimism or personality characteristics. It is one thing that I'm a little bit unsure what to make of that, actually, to be honest. And I think that part of it is the fact that it's a little bit of an opaque measure in that, yes, they differed from this temporal difference learning model. But, for example, I don't think there was any comparison to healthy controls in that. So it doesn't give us an indication of whether that was actually an abnormal response, or maybe the task itself was just not well characterized by that model. But it's a good question.
And I was interested on my end to see some of the data you presented on kind of the immediate responses to serotonergic medications in terms of amygdala processing and that sort of thing. Have you, I know you talked about the long-term effects in terms of changing how we view things and processing of cognitive information. Have you, in your own work, kind of noticed any immediate effects that you're now looking for if a patient started on an antidepressant?
Yeah, that's a great question. We tried to get at this a little bit in EMBARC, where we had the 1 week measurement point, and we looked at changes from baseline to 1 week, and we thought maybe we can pick up a early signal of predictive response. And at least for the emotional conflict task, we did not succeed and didn't find something that was a early change that might predict it. In my clinical experience, what I've seen is that usually people have the thing where sometimes they'll get a little bit worse before they get better with the antidepressant. It sometimes causes kind of an acute, almost exacerbation. But then, depending on the degree to which they bounce back from that or they're able to tolerate it, at least just from my perspective, seems to have some potential utility for predicting how they will respond to it. There's no empirical data here or anything, just simply a clinical observation. But I think that is a key question, which is, what is the acute effect? How do we isolate that from the therapeutic effect if we need to? And then how can we use that to try and predict the therapeutic effect?
Got it. Okay. Any thoughts on how all of this relates to bipolar disorder, bipolar depression?
Yeah, great question. I think the short answer would be that I wouldn't necessarily relate them. Okay. I think that there are some, commonalities in the neurobiological phenotype, but I think the mechanisms underlying the two are probably quite different. It remains to be seen, I guess, how, well, for one, I think I know a lot of psychiatrists will not prescribe a SSRI or an SNRI, for example, to somebody with a bipolar disorder, because they generally don't tend to work for treating the depressive elements of the disorder, which is actually the hardest part of bipolar, I think, really to treat. That would suggest that, there's probably different mechanisms going on that are provoking that, and the predictors might as well be very different.
Got it. All right. Well, just wanted to say thank you again for taking the time out to speak with all of us this morning, and appreciate you being here.
Excellent. Yeah, absolutely. Thank you so much for having me.
Okay. Thanks, everyone. Have a good day.
Bye-bye.